lecture video is all about protein synthesis, which is the making of proteins. Proteins do everything. Your DNA is basically a genetic instructional manual for making proteins. And so our goal during protein synthesis is to start with our DNA, which is our instruction manual, and then actually make or put together the proteins that our body needs to function. That is all that's going to happen here. There are two parts to this process, transcription and translation. And I'm giving you the very simplified version. Just know it's more complex than this, but we're giving you the high points. So, we want to make proteins, and we have this instruction manual that's found in our nucleus that's going to tell us exactly how to make specific kinds of proteins. But we have a problem. The cell has a problem. In all of its amazingness, there's one glitch that the cell has, and that is the DNA, which is the instruction manual for making proteins, is in the nucleus. And it is too fat, it's too big to get out of the nucleus to the ribosomes, which is where the proteins are made. So we have these instructions for how to put proteins together, but the place that's going to use the instructions to actually do that, which is the ribosomes, they are outside of the nucleus and they do not have access to the instruction manual. So the cell has to figure out a way in which to get the instructions out of the nucleus and into the ribosomes, where the ribosomes can then read the instructions and put the proteins together based on those instructions. And it's going to do this by using a special chemical or a special molecule called RNA. RNA is going to be the chemical link between DNA and the ribosomes. It's basically going to go into the nucleus, it's going to write down all of the instructions that are in the DNA, and then it's going to leave the nucleus and it's going to go to the ribosomes where those instructions can be used to put together a protein. Three types of RNA: messenger RNA or mRNA, transfer RNA or tRNA, and then rRNA, ribosomal RNA. We are only going to focus on messenger RNA and transfer RNA for our purposes. Okay, so rRNA sounds like DNA, right? And it is. It's very similar to DNA, but there are a couple of major differences. RNA is also a long chain of nucleotides, just like DNA. However, the nucleotides are a little bit different. It still has the phosphate group on the one end. It still has a sugar. However, the sugar is a different version of the sugar that is found in a DNA nucleotide. So DNA has the deoxyribose sugar. RNA has a sugar that's called ribose. It's still 5 carbons, it's just chemically different. And it also has nitrogen bases. It also has 4 possible nitrogen bases, but one of the nitrogen bases is different. So it still has A's, adenine, it still has cytosine, and it still has guanine, but there is no thymine in RNA. Instead, there's a nitrogen base called uracil. So instead of A, T, C's, and G's that are found in DNA, we have A, C, U's, and G, which are found in RNA. So if I gave you a random stretch of nitrogen-based letters and I said, is this DNA or is this RNA? Hint, hint, test question. Look for the T's and the U's. If there's a T, you know it's DNA. If there's a U, then you know it's RNA. So let's get through this process called protein synthesis. This is the process of decoding the genetic instructions, which are found on our DNA molecules, and using them to make proteins. There are 2 stages to protein synthesis: transcription and translation. Transcription is going from a DNA to a messenger RNA. That's what we're going to make. And that messenger RNA is small enough that it can leave the nucleus through the nuclear membrane and it can travel to the ribosomes and where the protein can get assembled. Translation is going to be using those instructions then to assemble the protein in the ribosomes. So, transcription, making a messenger RNA; translation, using that messenger RNA to make a protein. So here's how this works. We have a DNA strand right here. All right, we have unzipped it and we've unwound it basically. And the job of that is a chemical called RNA polymerase. It's going to come in, it's going to do kind of the same thing that DNA helicase did. It's going to unzip, it's going to unwind the DNA, and again, this is occurring all in lots of different places all on the DNA. and it's going to insert a messenger RNA strand. So here's how this works. This messenger A strand is going to be made by base pair rules again, except remember there is no thymine, there is only uracil. So A will bind with U and C's and G's will still bind together. So there's an A on this DNA strand. This will now be a U on the messenger RNA. So this is a messenger RNA strand right here. G, this nitrogen base on the messenger RNA is going to be a C. T will change to an A. C is a G. G is a C. A again, it's going to be a U because it's an RNA. T goes to A, C goes to G, and A goes to U. So we've inserted a new strand. We've put in the proper base pair based on it being a messenger RNA. So A's will bind with U's, G's will still bind with C, T's will go to an A, G will go to a C. Step 1, done. Now, we already talked about what RNA polymerase does, so that's going to separate the two strands of DNA, and it's going to insert this messenger RNA. Another important term to know is a codon. And a codon is just going to be a group of 3 letters, 3 nitrogen bases, on a messenger RNA. Okay, so UCA is a codon. GCU is a codon. AGU is a codon. And we're going to use these codons to basically put into place, we're going to translate these codons into an amino acid. So now that we've made our messenger RNA, and know that this is the simplified version, so there's a lot of different things that are happening to the mRNA between the end of transcription and the beginning of translation. Things just to kind of clean it up and get rid of junk letters and things like that. But for our purposes, this is really what I want you to be able to understand. It's just kind of the overview of what's happening. So we have finished stage 1. We went from DNA and we made our messenger RNA here. So now what we need to do is we need to use these codons, these series of 3 letters, in order to code or translation these into an amino acid. Proteins, which is what we're trying to make, remember, are long chains of amino acid. There are a whole bunch of different amino acids, but there are 20 that actually appear in our genetic code, and the arrangement of these will determine what a protein actually is, or what specific protein we have. So here's how this works. We're going to take our messenger RNA, and we are going to use the codons, the series of 3 letters on there, and we're going to code for an amino acid. So if you hopefully have your printed out sheet here, okay, here's the code for amino acids. You're going to want to print this out because you're going to need it for your tests, and it's nice and handy, all right? Look, we have UCA. That is our first codon. So here's how we translate this. So we're kind of translating into a different language, sort of. An amino acid language. So on this side, so on the very left side, you're going to find the first letter of your codon, okay? So we had, let's go back here, UCA, right? So U, here it is. So I know that I'm going to be in this row, all right? Along the top is your second letter, okay? So again, it was C, UCA. So find the C up on top. We got the U over here, so if I move over, the C comes over here. And then along the side, so I know I'm in this box, along this side here then, you're going to find your third letter. So it was UCA, here's the A. You're going to go all the way across until you find it right here, UCA, and that codes for the amino acid serine, S-E-R. What we would do, if we go back to our picture here, our messenger RNA, we know that UCA codes for serine. All right, so we're going to kind of make this little structure here, and we're going to put SER inside of it, because the serine is the amino acid. All right, so here is the amino acid, right? Now, let's look down at the next one. We have GCU, right? So again, look at your sheet here, okay? Find the G along the left-hand side, so here's the G. Find the C on the top, okay, that's the second letter, so we know we're going to be in this box right here. And then find the U over here, okay, that's the third one, and go across. So GCU codes for the amino acid alanine, ALA, right? So if we add that here, ALA, and then we have one more. So take a second, look at your chart, and see if you can find the amino acid for AGU. Pause this, see if you can find it, and then you can check to see if you're right. Okay, so A, find the A on this side, so we're in this row. G is over here, so we know we're going to be in this box over here. And then the U, if you go over, AGU, codes for the amino acid serine. So, on this strand of DNA, the instruction manuals to make this particular protein is a serine, an alanine, and a serine, in that particular order. All right? Now, here's the thing. We want to basically lock these into position. We don't want the alanine to like swap places with the serine by accident, because that's a completely different protein. Our protein that we are looking at is a serine with an alanine with a serine. So in order to lock them into place, we are going to use base pair rules again one more time. Okay? And we're going to lock it into place by connecting it using the base pair rules. There's a U here. This would be an A. C, G. A is going to go to U. I'll explain that in a second. G will go to C. G, A, U, C, A. And that is going to lock those amino acids in place so they can't move, and that is our protein. And so let's look at this whole structure right here. This whole structure is called a transfer RNA. And if we look at it like up close, okay, here it is. This is a transfer RNA. It's a structure, and this transfer RNA holds the amino acid and it attaches to the messenger RNA using 3 anticodons. These are the anticodons. This is the amino acid. This whole thing is the transfer RNA. So if we were going to sum up this process, the first step is to take the DNA and make a messenger RNA, a single strand of messenger RNA. Okay, base pair rules, no T's but U's. Then, that's transcription. Then we're going to take the messenger RNA codons, the groups of 3 letters, right? And we're going to use the chart to code for the amino acid that we need. So first letter on the left-hand side, second letter along the top, third letter on the right-hand side. Find the amino acid. We're going to put that in a structure called a transfer RNA. We're going to get them in the right order, and then we're basically going to lock them into place by connecting them to the messenger RNA using an anticodon, which is the other 3 series of letters found on the transfer RNA, so that those amino acids can't swap positions. I guarantee, look at your content assignments. Those are the types of questions you're going to see on the test, so you need to really make sure that if you have questions about how this is working, that you let me know. Have a great day.